Let me just go back through and pull out some last names here.
Also, you did not sound the way I thought you would.
What were you expecting?
Um, I know that you're from North America. I still didn't expect an American accent.
Oh, I didn't realize I had an American accent.
Well, you
I'm
sound very non-European.
see here our local type accent. It's very abrasive, so I try my best to do away with it. Like for example,
Mm.
the month that's coming up is gonna be September. No, 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 no. Uh, Alrighty, I think could be set. Mm. Sorry for the delay, everyone. You know how science goes when you have 36,000 copies of everything.
Well, we actually still have 6 minutes.
Oh, beautiful. Right. Ah, here we go. There's the one. Alt save. So Kraken, my screen share will go through your stream.
Yeah, so what will happen is I will be watching your stream and then streaming your stream.
Oh, a streamception.
Yeah.
Cool. Hopefully everything stays in sync.
It usually does.
Fantastic.
Wait, does your presentation have sound?
No.
Okay, excellent. That makes it easier.
No sound, no videos, no copyrighted information.
Excellent.
Everything that'll play nicely with the internet. Nah, Squib, I think we're just all sitting here.
Oh, well, we're supposed to start really soon.
Yeah, I think we're set for wheels up shortly. I don't know the standard protocol for a presentation here at the lovely BioCord. Um, but at least how I have been operating in the past um, is save your questions till the end. However, if there's something that I go over, that may be super familiar to me, but is unfamiliar to you, by all means, ask me to reiterate or clarify. That's totally fine. But the questions, we can really dig into them at the end.
Yeah, that's pretty much how they go. And so whenever you're ready, I can I will turn off my stream to Discord, and then you can show your screen and I can stream that to YouTube.
Cool. Yeah, let me just turn off all of my pop-ups and fly-ins. Think we should be good. All right. I think I have to go full screen first and then stream. Sure. Give you guys some good quality. Source and go live. There's the Discord. Can you see my white screen? Preferably with the words in it.
Yeah, I can see that.
Cool. You turn on my laser pointer then. This guy's all good too.
Yep, laser pointer's good.
All right. Awesome. Well, we I have wheels up, Kraken.
Yep, stream is up on YouTube as well.
Fantastic. Well, hello everyone. My name is Chris, or Dr. Or my lab name C Money. And today I'm going to be talking to you guys about my work that I've been doing, wrapping up, and subsequently transitioning away from, and that is a functional analysis of virulence in Vibrio Coraliticus RA22 and probiotic mechanisms of Phallobacter Inhibins S4. And I think today's date is the 27th of August, 2021.
It's the 28th.
It is the 28th. Note, today is still correct. So I have three topics that I want to dig into today. The first is an investigation of the two type 6 secretion systems in Vibrio Coraliticus RE22SM and how they exhibit differential target specificity for bacterial prey and oyster larvae. The second topic will be the role of quorum sensing in Vibrio Coraliticus during oyster infection. And the third topic will be the potent probiotic potential of Phallobacter inhibins S4 and its probiotic activity in an aquaculture setting. So, I want to start with some background on a half shell for you guys. And the organism and the model organism I've been working in is bivalves, more importantly, bivalve aquaculture and specifically oysters. So, bivalve aquaculture is not limited to oysters, but that includes anything that comes in a shell: clams, mussels, oysters, scallops. And here in the United States, they are an important sector of agriculture. They were valued at $441 million in 2018. And of that, oysters accounted for over $280 million of revenue. Here in the US, we consume almost 60% of the total oyster production. And in my little state, they are the dominant aquaculture product, with over 8 million of these guys harvested in 2017. Now, there are two dominant species of oysters in aquaculture. There's the eastern oyster, Chrysostrea virginica, and the Pacific oyster, Chrysostrea gigas. I work with the eastern oyster being on the East Coast. And now, besides their economic importance, these oysters provide habitats for other estuarine species, and they will filter feed to remove planktonic organisms from the water, really cleaning it up and making it a happier place. Although, once they do that, you don't want to eat them. So down here on the left is a typical aquaculture hatchery setup where there are these rearing tanks that hold the small oyster seeds and larvae. And after a certain amount of time, they begin to settle down and they will adhere to the tanks or artificial structures assembled in those tanks to sit down and grow into these oysters that you can buy at a restaurant about 5 years later. So now I've kind of discussed why I'm doing this and what we're looking in. Now I want to go a little deeper into the organism. So the pathogen of interest here is Vibrio Coraliticus RE22. And from here on out, I'm going to call it RE22 because Coraliticus is a mouthful. It is a member of the gamma proteobacteria. It's a gram-negative facultative anaerobe it's a curved rod morphology with a singular polar flagellum, as you can see down here in the bottom right. And this strain causes vibriosis in larval and juvenile oysters. So with my work, I'm focusing on these oysters when they're little itty bitty, not full size, but they're about the size of a grain of sand. So RE22 has a suite of potential virulence factors to really specify, its target and really do the job it needs to do quickly and efficiently. So it contains a host of secretion systems, including type 1 secretion for an RTXA Martax toxin, type 2 secretion for the cytoplasmic protein export, type 3 secretion for all the nastiness that type 3 secretion does, and the focus of my talk today is the type 6 secretion. Additionally, there is metalloprotease activity, homolysins, and more virulence factors to be discovered. I had the virtue of this organism having its genome sequenced, and it breaks down into 2 chromosomes and a megaplasmid. Our lab did this back in 2015, and then it was confirmed by folks at the USDA in 2018. So we could take our sequences and close up the genome thanks to the other folks, Richards et al. So I'm going to jump right into it. The first manuscript that I want to talk about is the 2 type 6 secretion systems in Coraliticus and how they exhibit differential target specificity for bacterial prey and oyster larvae. And this is a contact-dependent injectile apparatus. And this paper is currently in revision with the folks at Hemisphere. So what is the type 6 secretion system? I've said it a bunch. Some of you might not know what it is. Well, what it is a bacterial nanomachine for protein and effector translocation and delivery. First thought for interbacterial antagonism. And this specific type 6 was first identified in 2006 in Vibrio cholera. And how it works quickly is this base plate complex made up of TSSM, J, L, first recruit to the peptidoglycan layer when beginning the assembly. Next, the base plate complex here, TSSA, EF, GK, fully assembles and then recruits TSSB and C, which encompass the full injectile apparatus, which is HCP. It's a stacked hexameric ring structure to translocate out of the cell. It on top can have a decoration of this VGRG protein, which adds a sharpened tip, if you will, through protein tetramers. And then lastly, it can have a PAR motif, which is, you can think of that like an armor piercing bullet, the tip that goes on there. This apparatus fires rapidly and is conservatory in the fact that if it infects a sister cell or another Vibrio, it can recycle these components, and they don't necessarily function as effectors for pathogenic effect, but effectors for a sister cell activation for more type 6 firing. And it is ATP dependent through a ClipV apparatus. And what's interesting about this is, much like a trebuchet, it doesn't take energy to fire more so than it takes energy to reset this mechanism. And it fires really fast. I think the polymerization rate of the HCPs is somewhere around 14,000 RPMs. So now in our organism, I was talking about, cholera and the historic findings previous. Our organism contains two type 6 secretion systems with the potential for divergent function. So in cholera, it has one type 6 system with two functions, and in the soil pathogen Burkholderia thailandiensis, that has five type 6 secretion systems. So by virtue, I got a simpler system to work with, and I think it gets really interesting from here on out. So the first thing I did was I mined our genome, just doing some direct analysis, seeing what in the world was there, what was present. And I found some complementary results. On chromosome 1, we had an HCP, this is a hemolysin co-regulated protein, a VGRG, a valine glycine repeat protein G, and an upstream PAR containing protein. And what PAR stands for is proline, alanine, alanine, arginine, and this functions for effector decoration and hardening. On chromosome 1, this gene neighborhood for type 6 spanned 22 genes encompassing just north of 27 kilobases. On chromosome 2, I found similar domains and homologous proteins, HCP2, a VGRG2, and what made this chromosome 2 stand out was there was this endopeptidase domain, which targets components of the peptidoglycan, and it contained a Lys-M motif from Pseudomonas aeruginosa. And on chromosome 2, this type 6 gene neighborhood contained a larger genetic contribution of 27 genes that spanned a smaller amount of 25 kilobases. So these findings here suggest that the type 6 secretion system 1 on chromosome 1 may be geared toward attacking eukaryotic hosts, whereas the type 6-2 appeared to be for bacterial antagonism through this endopeptidase domain. So then I went a little bit further. There are these folks out of Tel Aviv University in the Dor Solomon lab that were doing more of a computational bioinformatics approach, looking at mix motifs, which are markers for type 6 effectors. And these guys are conserved motifs within genes often located near the type 6 gene neighborhoods. And they elucidated three motifs: a conserved motif that's more centrally located within the protein, as well as a less conserved N-terminal and C-terminal motif. So I focused on the conserved motif here, with H standing for hydrophobic amino acids, X is anything. And what I found was that there were no mixed motifs associated with type 6 secretion system 1, which I thought was interesting because we have these genes encoding these proteins for pathogenic effect. However, there were a bunch of mixed motifs associated on type 6 secretion system 2. We had a Vas-J protein, which is virulence-associated secretion, and here's the motif. And that was conserved and terminal domain of an IMP-A protein for export, an essential component of pathogenic effect. Yeah, that's the best annotation we get. Next was a TSSA protein, which is an IMP-A homologue for protein processing, peptide secretion, and trafficking. Again, really leaves much to be desired. But when we go one step deeper, we found this VGRG2 protein, or TSSI, that contained two mixed motifs off of that central conserved motif that were kind of juxtaposed at either end of the protein, but not at the terminal domains. And this protein here contained three motifs of interest, one being an RHS, a rearrangement hotspot for the VGR protein, which contains DNase activity. And VGRG itself contains a phage tail assembly protein to allow this to assemble and export, as well as a GP5 superfamily, which is for that base plate hub subunit and tail lysozyme, so the type 6 secretion system can exit the cell safely without damaging the attacking cell. So that's all fine and well. I found this in my organism, but what else is there? What else is associated with a type 6 secretion system? Well, there's a bunch. So it breaks down into two families, one being the complex loading effectors. So you can have toxins that fuse to VGRG or PAR, proteins binding the inner HCP tube, or proteins that interact with VGRG or PAR but aren't fused directly. There are also a slew of families, effector families associated, with the peptidoglycan hydrolases and phospholipases being the most well studied. There are still nucleases, actin cross-linkers, ADP ribosylases, and pore formers present. But again, when we break this down into a little pie chart, more than half of what's associated with the type 6 are just transmembrane helice-containing proteins. That's all we have. And more than a quarter of these are still unknown. So now that I've gone ahead and set this up and looked at some computational approaches, I wanted to see how well my organism and its type 6 secretion could kill just a regular old lab strain of E. coli. So here's my standard type 6 secretion assay, where I have my attacking cell RE22 and my prey cell E. coli SM10. And these guys are set up in a ratio of 4:1. So there are 4 attacking RE22 cells to 1 E. coli cell. So that roughly works out to a starting CFU of 2 times 10 to the 9 for RE22 and 5 times 10 to the 8 for SM10. And what we saw over the course of 4 hours in this experiment was the attacking RE22 went up a little bit. Just a little bit. However, the E. coli got decimated. They dropped more than 3 logs over the course of 4 hours. And in my tenure, I have done this experiment close to 100 times, and we always see this drastic drop with the E. coli. So now, how do various mutations in the type 6 secretion system affect its ability to kill E. coli SM10? Well, we're going to look at it. So here in figure A, I broke this up by type 6 secretion system 1 on the left and type 6 secretion system 2 on the right. So here we are just looking at the E. coli numbers because no one really cares about the attacking cells that don't change, and it makes the graphs really muddy. So what we see here is that when E. coli is attacked with RE22SM for 4 hours, they drop, about 3.5 logs. When I mutated the HCP1 in Coraliticus, I saw an attenuation of virulence where they were only dropping, a log or so over 4 hours. Whereas when we knocked out VGRG1, the tip of the type 6 apparatus, we saw complete virulence and there was no statistical difference compared to the wild type. In figure B, we can see the same killing level with the wild type Coraliticus against E. coli. And when I knocked out HCP2, they were completely attenuated. There was no significant difference between the starting inoculum of E. coli and the recovered CFU after 4 hours. However, when we knocked out VGRG2, we saw that there was, again, no difference in virulence over the course of 4 hours. So the take home from this is that the loss of HCP1 or HCP2 results in a complete or partial attenuation of virulence, whereas the loss of VGRG1 or VGRG2 had no significant impact on virulence, and they maintained their ability to kill with that type 6 secretion system. So next, I went ahead and made double mutants. I knocked out both HCPs and both VGRGs in different mutant backgrounds and did the same experiment. And here with the wild type Coraliticus attacking E. coli, we saw that same 3 log drop over 4 hours. However, HCP1 and 2, when you knock out both of them, does nothing. It's like we didn't even add them. Um, there was no change over 4 hours. Whereas when we knocked out VGRG1 and 2, we did see an attenuation in virulence, but there was still killing going on. And this makes sense because you still have that injectile apparatus, the HCP. You just don't have the tip sharpened nicely. And what we can take home from this is that loss of HCP really impacts the ability of the type 6 secretion system to assemble, fire, or deliver its pathogenic components. So now, how do these type 6 secretion mutants affect the ability to kill our model system, our larval oysters? So in the larval oyster experiment, what I will do is I will beg with oyster hatcheries to send me oyster larvae. And when they do, they come, they're about the size of a grain of sand, and I will take these and dilute them down into a 6-well dish, shooting for about 100 oyster larvae per well. Then I will inoculate them with the pathogen for 24 hours, and I will begin quantifying my experiment when the wild type, the second bar here, reaches killing levels to about 40 to 60%. So here, this first bar is the no treatment control, just oyster larvae fed and kept in seawater. They have about 95% survival. When challenged for 24 hours with the Coraliticus, we see them drop in their survival to about 50%. And here, when I challenged with the HCP1 mutant, there was no difference compared to the no treatment control. It's as if I didn't even add them. And same result with the VGRG1. And this right here, this really speaks for itself that if you mutagenize components of the type 6-1 that we previously hypothesized as involved in oyster virulence, we see that they're attenuated, they're avirulent, these oysters really don't care. However, going forward, looking at the HCP2 and VGRG2 components of the type 6-2, we saw that when you knocked out HCP2, there was an attenuation of virulence, and that attenuation was even more pronounced in the VGRG2 mutant. So then the type 6-2 mutants are only attenuated in their virulence and ability to kill as compared to the type 6-1 genes. And then lastly, I wanted to look at the double mutants and how they behaved. And here we saw some partial killing ability restoration in the VGRG 1 and 2 double mutant, while the HCP1 and HCP2 double mutant was not different than the killing ability of the HCP2 single mutant. So the take-home conclusions from this component is that Vibrio Coraliticus RE22 contains 2 type 6 secretion systems, where type 6-1 and type 6-2 have primary targets, and they both contribute to these secondary targets. And RE22 HCP mutants are severely attenuated for type 6 secretion system, indicating that HCP is essential for type 6 firing, and that a functional HCP is more important than a functional VGRG. A needle will still work if the tip isn't super sharp. And lastly, that HCP1 and VGRG1 are important for eukaryotic virulence as their primary target, and that HCP2 and VGRG2 are important bacterial virulence factors. Now, we're going to change role. We're going to change it up a little bit, switch some gears, and we're going to jump into the role of quorum sensing in Vibrio Coraliticus during oyster infection. And this is a density-dependent virulence factor. And I'm currently in the process of writing this up, making pretty graphs, and I'm gonna submit it off to Applied in Environmental Microbiology. So, what is quorum sensing? Some of you may know, some of you may not. Well, here we go. So, quorum sensing is the production of intercellular communicatory molecules that are produced at a cellular level. So, these guys aren't controlled, they are not controlled by gene transcription or any sort of regulation like that. It's one cell will make one unit functionally. And what are these communicatory molecules? Well, they're called autoinducers. And here it's AI1, which is an N-acyl homoserine lactone, or I'm going to call it an AHL. Here's the basic structure. With this R group, it can be saturated to different degrees and have a different acyl chain length. So we can look at this in two ways, either a high cell density state or a low cell density state. So here I'm going to describe low cell density, where there aren't enough cells to create enough AHLs in the local environment to tip this scale of Lux N. And what Lux N is a dual function histidine kinase phosphatase sensor that, in the absence of adequate AHLs, will phosphorylate this system and will function as a kinase. And will do that through the action of the phosphotransfer protein LuxU to the phosphorelay protein LuxO. Phosphorylated LuxO will then recruit sigma 54 to recruit and transcribe these 5 QRRs, which are quorum regulatory small RNAs. And those with HFQ will ultimately block VCPR transcription. And VCPR is a LuxR homolog for those of you familiar with the Vibrio Harvey system and bioluminescence in bobtail squid. So if you have an interruption or degradation of the VCPR transcript, your organism will be in a low cell density state, favoring a holding pattern, such as biofilm formation, waiting for the numbers to increase. So the take home here is that if you have a low concentration of autoinducer, you're going to have a low concentration of the VCPR transcript, subsequently the protein, and lower levels of virulence. So to address this, I generated 3 mutations, one in LuxM, the AHL synthase, LuxN, the sensor, and VCPR, that master regulator. Now, on the flip side, in a high cell density environment where you have enough cells producing enough autoinducer into the local environment, it will then go ahead and bind to the LuxN sensor, switching its function from a kinase to a phosphatase, subsequently draining the phosphates out of the system. So LuxU phosphate goes to LuxU, LuxO phosphate becomes LuxO. And unphosphorylated LuxO does not allow for the recruitment of sigma 54 or the transcription of the QRRs or the involvement of HFQ. So you go on and you get transcription of VCPR. And in this high cell density environment, it tells these cells we're primed, we're ready, we're going to fire, we're ready to fight. And in my organism, this is indicative of an increase in extracellular protease activity. And to mimic this in a cell mutant background, I mutagenized LuxO. If you don't have LuxO, it can't be phosphorylated. It can't be phosphorylated high cell density. So now, how do quorum sensing mutations affect Coraliticus? Well, there has been previous work looking at VCPA and VCPB transcription, which these guys directly influence metalloprotease activity, which are controlled by VCPR, this LuxR HapR homolog for the master regulation of quorum sensing. And lastly, some folks at the Oregon State University discovered that extracellular metalloprotease will directly inhibit another virulence factor, the extracellular hemolysin. So we're thinking we knock out metalloprotease activity, potentially see an increase in hemolytic activity. So now, how do various mutations in quorum sensing affect protease activity? Well, what I saw in my protease assay, which was I was utilizing a colorimetric assay of casein, azocasein degradation. So over the course of 4 hours, I will just call you to the 4-hour spot. Over 4 hours, I saw no difference and no change in the LuxO mutant, which we hypothesized to be a high cell density mimicking mutant, compared to the wild type. Whereas the LuxN, VCPR, and LuxM mutants were pretty much stacked on top of one another at 4 hours, making approximately a third of the protease that the other two strains were producing, and this can be attributed to, the cells have other proteases. Not everything's pathogenic. We couldn't adequately address all of these components, but we did see a stark reduction. Next, so how does a complete quorum sensing circuit influence biofilm formation? So on the left here, I did a biofilm assay where I grow my cells in a 6-well dish containing a sterile cover slip, and I addressed the static planktonic growth in the biofilm assay here in figure A. And what we saw was that LuxN and VCPR were no different than wild type in their ability to grow under these static conditions, whereas LuxO and LuxM were reduced over the course of 24 hours. And then in the biofilm formation assay here, where I took the cover slips and quantified the biofilms off of those, we saw that there was no difference in the LuxN, VCPR, and LuxM compared to the wild type in their ability to form biofilms. Whereas the LuxO mutant was actually increased, which was not what we initially hypothesized, but it was interesting to go forward. So now next, to fully address this, I'm going to jump back to the type 6 secretion system and see how the quorum sensing state and quorum sensing system influences type 6 mediated killing of E. coli SM10. So here, this is the standard type 6 assay that I described in the last component of the talk. And what we saw in this experiment was, now this is just the the 4-hour numbers for E. coli, and these strains are noted at the bottom. In a mutant background deficient in the LuxN gene, encoding for the AI1 sensor, we saw a complete attenuation in virulence. There was nothing going on there. Whereas the LuxO and VCPR mutants were slightly attenuated over the course of 4 hours, and the LuxM mutant, that AI synthase, showed no difference compared to the wild type. It was unaffected and was able to fire as if nothing had been changed. So now, lastly, does this quorum sensing state in RE22 contribute to oyster mortality? Well, the answer is yes. So here we have our no treatment control oysters coming in somewhere at 95% survival. Our wild type strain was killing at about 50% over the course of 4 hours. When challenged with the LuxN mutant strain, our survival drastically increased compared to the wild type, coming in at about 80% survival, whereas the LuxO mutant showed no difference compared to the wild type. The only difference was bigger error bars. The VCPR mutant, that master the master regulator, was again attenuated for virulence or increased survival. And same with the LuxN, that AI1 synthase was attenuated for virulence. And two mutants that I hadn't previously discussed, but threw in here, are the VCPA and VCPB genes that directly translate the metalloprotease. VCPA creates the pre-protease, VCPB processes that and secretes it for proteolytic function. So what we can take home from here is that quorum sensing does impact oyster survival through these VCPA and B genes, and we weren't able to knock out, a LuxO mutant and increase virulence over the course of this 24-hour experiment. So again, conclusions is metalloprotease activity is linked to the appropriate mutation-induced cell density state, either high cell density or low cell density. Biofilm formation in these quorum-sensing mutants was relatively unaffected, where the planktonic growth was overall decreased, and these low cell density quorum-sensing mutants improved larval oyster survival, and the high cell density quorum-sensing mutant LuxO killed oysters at wild-type levels. Nothing interesting to report with that one. Now we are going to drastically change gears stop thinking about the stop thinking about the pathogen, and we're going to look at the probion. So we're going to jump into Phallobacter inhibins S4 and its probiotic activity in an aquaculture setting. And this is more of a functional analysis of quorum sensing and probiotic activity. So, what is Phallobacter inhibins? Well, I got you. It is a probiont that was isolated from the inner shell surface of a healthy oyster. It produces a novel antibiotic called tropodiathletic acid, or TDA. It's a robust biofilm former. It forms these hallmark rosettes, and it contains a PGAIR model of quorum sensing that is an ortholog to the Vibrio fisheri Lux IR system. And it communicates through N-acyl homoserine lactones that not only serve as a quorum sensing molecule for S4, but they are quorum quenching and inhibitory for Vibrio coraliticus. And here's just a brief model. Not much is known about the quorum sensing system in Phallobacter, but what I can tell you is that the PGAI gene generates that AHL that is then detected by the PGAR gene. Then the PGAR gene will control the target genes for one, TDA synthesis, as well as for AHL production. So, in the right environment, this actually becomes a positive feedback loop where you make AHLs to make more TDA, which subsequently makes more TDA and AHLs. So, you can get a really good snowball effect in this model. Now, we have previously demonstrated that these AHLs from S4 will inhibit the metalloprotease activity of Vibrio coraliticus. And here are the three AHLs with their full chain lengths shown. And what we see here is the compound DR8 shows the strongest inhibition of protease activity in Vibrio coraliticus, whereas DR12 and DR11 aren't that far behind. They're all doing something. And again, these were isolated compounds, isolated fractions. And when you take these together, it is an additive effect. So now I wanted to see if I could sort of address this um, in a mutant background rather than digging into the chemistry. I wanted to see if I could create a mutant that would better inhibit Vibrio coraliticus. So first thing to do was to see what AHLs were being produced by my Phallobacter mutants. So first, I have to apologize. This is the best output I could get off of our LC-MS machine. So just bear with me. What we have here in figure A is I'm looking at the C10, so 10 carbon length AHL, and that was produced in trace amounts in my mutant of PGAI, the AHL synthase, as well as PGAR, that receptor protein. In the Lux, I generated a Lux O mutant, which is again a phosphorelay protein. And here the C10 AHL was produced at 40% of wild type. And lastly, the C10 AHL in a PGAK mutant, which is a histidine kinase that I identified, was being produced at 60% of wild type levels. So all around, the C10 AHL was knocked down in my mutants. Whereas in the C14 AHL, the much longer and more substituted chain length, this was undetected in the PGAI and PGAR mutants. And at this particular time, so 6 minutes, 6.5 minutes, it was only detected in trace amounts in the Lux O mutant, as described by that very wimpy light blue peak. And this is where it gets interesting because the C14 AHL was produced 2.25 fold higher in the PGAK mutant. You can see this gray bar or gray peak here compared to the dark blue of the wild type. Now, previously I showed 3 AHLs. Well, only 2 of them were detectable under these culture conditions. The previous experiments required us to grow up upwards of 80 liters of culture supernatant and purify from that, whereas the LCMS-MS lets me do it with 10 mils, which is a lot better. So next, that's a wrong slide. We're going to jump into biofilm formation and look at how well these Phallobacter mutants form biofilms. Now, first, I wanted to address their planktonic growth because these guys are obligate aerobes and they do very well in a highly oxygenated, turbid ocean environment. Well, in static conditions, we saw a reduction of growth in the PGAI and PGAR mutants, whereas the LuxO mutant here was growing at wild type levels and PGAK was increased over 24 hours. So now for biofilm formation, we see an even more pronounced trend, whereas the PGAI and PGAR mutants form a very weak biofilm. Between 10 to the 10 to the 3, so there were like 200 cells on a typical glass cover slip, which is insane. And next, the LuxO mutant was again producing biofilms at wild type levels, and the PGAK was significantly increased in this experiment, indicating that maybe this PGAK was onto something, or it's going to potentially lead to another probiotic for use in an aquaculture system. So then, really, I keep coming back to the type 6 secretion system and testing it because it really seems to be something important. And I wanted to test how well these mutants were able to resist the type 6 mediated attack of Vibrio Coraliticus. So again, I'm just showing the Phallobacter prey numbers over the course of 4 hours. And these statistics are compared back to the wild type here. The PGAI and PGAR mutants, as well as this TDBD mutant, TDBD is an end processing gene for TDA biosynthesis. So when you knock out PGAI, PGAR, and TDBD, you see that S4 is most susceptible to this type 6 mediated attack. Whereas ClipX here, which is again just an ATPase, that we found linked to TDA production. No ClipX, no TDA being produced. This strain was more susceptible, but not the most susceptible. And what's interesting about these 4 strains is that the PGAI, PGAR, ClipX, and TDBD are all deficient for TDA production. So now the LuxO mutant and the PGAK mutant showed no difference over the course of 4 hours compared to wild type. And these guys are TDA positive and TDA producing. And lastly, the most interesting result here was this XOP mutant, which is deficient for biofilm formation. This is, for the exopolysaccharide synthesis. And this guy fared the best because when I set up this experiment, I will take the attacking and prey cells and I will filter them onto a 0.22 micron filter. So I'm creating an artificial biofilm. However, this strain doesn't behave classically, and other layers of the biofilm don't shut down. They're all active. They're still all cranking out this TDA. So what we can take home from this type 6 assay is that the direct or indirect TDA production will directly influence survival in a type 6 assay. And now lastly, how do these type 6 quorum sensing, how do these S4 quorum sensing mutants improve oyster survival when challenged with Vibrio Coraliticus? So this chart has a little bit more data in it than the other oyster challenges I've showed you, so I'll walk you through it. The first is the no treatment control, just oysters, seawater, and food. Here is the no treatment and the addition of Phallobacter S4. Again, no difference compared to no treatment. Our wild type here, our wild type S4, Coraliticus was killing at about 50% over the course of 24 hours. And when we pre-treated with S4 and then Vibrio Coraliticus, we saw a drastic increase in survival, upward close to 90% compared to less than 50%. And then when we added or pre-treated with the PGAI and PGAR mutants, we saw an increase in survival. Not necessarily because the Phallobacter mutants here, PGI and R, were doing anything, but there was just something in the way of the Coraliticus. Either potentially, these S4 were functioning as food sources or just disrupting the Coraliticus. It doesn't matter. The oysters survived better, which was awesome. They're protective, but less protective. Next, when I pre-treated with the Lux O mutant, saw no significant difference compared to the wild type challenge. And then lastly, when I pre-treated with the PGAK mutant, we saw protection levels at no treatment levels. So basically, this last bar here is no different than the no treatment that had no bacteria added at all. So this completely mitigated the effects of the Vibrio Coraliticus and its effects on survival of these larval oysters. So to conclude, wrap it all up, it's that TDA production, biofilm formation, and AHL biosynthesis are essential for probiotic function. And I mentioned it a couple times while going through, but you need to pre-treat with these organisms, with the S4. All larvae survive better with the S4. And they're present, they have to be present before infection or before the challenge to really work well. We've done experiments in the past where we co-treated, the pathogen and the probion at the same time, and the pathogen won every time. And then lastly, this PGAK, which is a wall K homolog from Staph aureus, improves probiotic potential. And note that we had both the PGA-IR quorum sensing system and a wall KR type system present. So just to sum all this up, it's that in my project 1 was that HCP is essential for type 6 mediated virulence. It's an important virulence factor, the hemolysin co-regulated protein, and it allows for VGRG translocation. For 2, quorum sensing is essential for metalloprotease activity and virulence, and quorum sensing has an interesting effect on type 6 mediated virulence, especially through the LuxN mutant, indicating some potential linkage between quorum sensing and type 6. And then for my third project is that a full probiotic suite is required for Phallobacter Inhibins S4 protection of oyster larvae or just direct inhibition of Vibrio coraliticus. And mutations in PGAK unilaterally increase TDA production, biofilm formation, and AHL synthesis. So future directions. Well, if I could keep doing this for years and years and years, what would I do? I would keep investigating all of these because you need to understand the probion to understand the pathogen, and you need to understand the pathogen to understand the probion. What is this? Is this VGRG for type 6 associated with DNase? Is that a novel virulence factor? Do type 6 mutants affect the injectosome transcription overall? And what would effects of reciprocal mutants be on virulence if I were to knock out HCP1 and VGRG2 in the same background? What would we see? For quorum sensing, I would want to know what the role of APHA is. And APHA is a protein that acts reciprocally to VCPR. So when VCPR is up, APHA is down. Next, what's really interesting is that could this LuxN mutant be that linkage between type 6 and quorum sensing? Only one way to find out. And then next, how do these quorum sensing mutants, how would they affect type 6 secretion? Or excuse me, type 6 transcription? And then for the Phallobacter and the manuscript 3, I wanted to see what are the other genes present in that wall KR regulon? What are the roles in S4? And how does S4 impact RE22 virulence factor expression? So I want to say thank you to you guys for tuning in, and thanks to all my lab mates, Dr. Dave, the probiotics group we work with, Jackie, Nate, Kira, Damien, USDA. Those folks are giving me money to waste plastic and hurt the environment and learn something, and the University of Rhode Island for giving me space to park my butt and do this science. So with that, I will conclude my talk and open it up to questions for you guys. So thank you.
So I did ask a couple of questions, and I think other people might have some questions.
Yeah. So I'm not sure the best way to do this. I'm just going to keep it open and read the questions and then go through. Oh, wow. Oh, yeah, I muted. I didn't hear you guys. That was awesome. Okay. Uh, so fun fact, yeah, Lolly, we work with Vibrio parahemolyticus, and that thing is awful. It grows so fast and it smells so bad. I don't want anything to do with it. I'm sorry about an ear infection. All right. This is following out. Okay. Yeah. Aqua here in Rhode Island. We have a lot of estuaries, and those have really been capitalized on. Um, we crank out a lot of oysters here. So these oysters do not make pearls, and it's a shame because I have had to shuck thousands of them, and not a single pearl. Yes, and the oyster larvae, they start as seeds, which are think of them like seeds. And oyster larvae are when I get them, they're about a week old, and they're very interesting to wrap your head around because you put them into this seawater and they just suspend themselves like little floating grains of sand. It's crazy to think why aren't they sinking down? But after about 3 weeks, they do settle.
How are they collected?
So how they're collected is let me can I start? I'll just jump back to the beginning because this will make it a lot easier. Is here these tanks? They have to do frequent water changes. So when they do these water changes, they just drain everything through a coffee filter or a series of filters of different sizes. So yeah, they filter them out and they'll mail me. Basically, it looks like a coffee filter with used coffee grounds in it.
So if they don't, what do they usually do with the larva?
They will usually let these larvae grow up, and so they will test them and use them for more of a quality control to make sure they are on track for their aquaculture product. Then they will either be left to grow in this aquaculture hatchery system for mature oysters. Again, this is a larval setup I have in the bottom left, but there are mature oyster systems that are done indoors, or they are distributed to a more natural, wild oyster hatchery. We got other quorum sensing people here. Fantastic. All right. Do you guys have any other questions? Like, I'm all for it. There's no such thing as a dumb question. Can we unmute him, Kraken?
Oh, yes. Who am I unmuting?
I don't know. I suppose it was more of a if we can rather than who's ready.
Yeah, they can be unmuted.
Yeah. I know I glossed over a lot of points because I wanted to get through everything. But if you guys have technical questions or hell, even questions about how I got where I was, the rest are self-made. Okay. Um, yeah, and like a little bit more about me is I finished up my work and I'm drastically transitioning over to dental research. I'll be researching novel oral microbes and their implication in periodontal disease starting next week.
That's very different.
Radically. Oh yeah. I'm going from functional experiments and you know aquaculture systems to I'll be glued to scanning electron microscope.
I think we tend to have a very silent group.
That's all right. As long as my words all came out in the right order, that's totally fine.
Well, if there are no more questions, thank you so much for this talk.
Oh yeah, you're welcome. Thank you for tuning. I was not expecting this turnout. Thank you so much, guys.
Oh yeah, we
Happy to
also
share it with
had
you.
a couple more on YouTube, and I know a bunch of other people also expressed that they are not able to watch right now, so they were going to watch later. And so they might have some questions for
Yeah.
you.
They can just message them or do they ask them in the lectures and conference thing, or they can totally just message them right to me?
Both will work. Um,
Yeah, either one, because I know, like, if somebody else has that question, we can leave it, or if they want to private message me, that's totally fine.
yeah, they could do it in the chat. That is okay. But yeah, thank you so much.
Cool. Thank you guys, and have an awesome day.